Today we'll be talking about sex determination. Sex is determined when a zygote is formed as a product of an egg reaching the ovum, creating a zygote. The zygote inherits 23 chromosomes from each parent, making a total of 46, and also making it a diploid. An allele is forms of genes on chromosomes or different variations. Let's talk about chromosomes. Autosomal chromosomes are the first 22 pairs of chromosomes on the zygote. Sex chromosome constitutes the 23rd chromosome. Genomic imprinting is a definition for epigenetic changes in the zygote that result in silencing of the allele derived from one parent, leading to the expression of only the non-imprinted allele of the parent in the offspring. This has to do a lot with the X chromosome, which is why there are many mutations and diseases involving the X chromosomes. The set of chromosomes on a female is 2 X's, represented by 2 X's, and for males is represented as XY. In a male, the XY chromosomes will develop into Wolffian ducts or mesoniferous ducts. Around day 60, this will secrete the Mullerian inhibiting factor by the Sertoli cells. This Mullerian inhibiting factor is the factor that prevents Mullerian ducts from growing and making a female. Secretion of testosterone by the Leydig cells causes growth and development of the Wolffian ducts. This in turn makes or develops into male accessory organs. For the female will develop Mullerian ducts or paramesonephric ducts. Female accessory organs develop in the absence of testes, not as a primary development, and the absence of testosterone will make a female. Let's discuss the male reproductive system. The epididymis receives tubular products including spermatozoa and tubular secretions. The vas deferens carries sperm from epididymis out of the scrotum and into the pelvic cavity. Spermatozoa enters at the head, quote-unquote, of the epididymis and is drained at the tail, quote-unquote, by this structure. The redded testes is a tubular network that connects seminiferous tubules at both sides. Seminal vesicles add secretions that pass through their ducts, and the vas deferens becomes the ejaculatory duct at this point. The prostate secretes fluid through pores and walls of the urethra that nourish and protect sperm. Finally, the urethra carries urine from bladder to the outside of the body. Let's go ahead and talk about the interaction between the hypothalamus, anterior pituitary gland, and the gonads. The hypothalamus works as a sensor, integrating center, and an effector all in one. The hypothalamus is where the gonadotropin-releasing hormone is released. This is being released into the anterior pituitary, which is also a sensor, integrating center, and an effector all in one. The anterior pituitary gland is responsible for releasing the follicle-stimulating hormone, or FSH for short, and the luteinizing hormone, or LH for short. These two hormones constitute gonadotropins and are secreted into the gonads. Now, the gonads can either release inhibin or a sex steroid. Inhibin is a hormone that functions in negative feedback control of the follicle-stimulating hormone secretion. These sex steroids can be sent to alarm the anterior pituitary gland and also the hypothalamus, creating a negative feedback response. The gonads, when they release inhibin, will create a negative feedback response into the follicle-stimulating hormone. After the gonad receives these signals, this will create a gamete, or the sperm or the ova. Let's move on and discuss spermatogenesis, which is the formation of viable spermatozoa. At the start, there are 2 diploid cells, or 2N, meaning they have 23 chromosomes from the mother and 23 from the father, that will start off and produce spermatogonia. Spermatogonia are spermatogenic stem cells that migrate from yolk sac of testes during early embryonic development. They happen to be diploid cells which become haploid gametes by meiosis. Meiosis occurs and turns this diploid cell, which is called the primary spermatocyte, into two separating haploid cells called the secondary spermatocytes. This occurs via the first meiotic division cycle. At the second meiotic division cycle, these two haploid secondary spermatocytes will divide and produce two haploid spermatids each, creating a total of 4. After that, these spermatids will undergo spermatogenesis and form 4 viable spermatozoa. So one spermatid will form 1 spermatozoa. Let's talk about male fertility for a little bit. Male fertility. On average, the volume of semen per ejaculation is 1.5 to 5 milliliters. On average, for every ejaculation, there are about 60 to 150 million sperms. Also, male fertility decreases as the age increases. A vasectomy is a procedure which involves cutting or tying of the vas deferens and replacing it with a similar valve instead.
Okay, so we're going to be talking about the female reproductive system now. Some of the basic organs are the ovaries, which is the female gonads, and it's the site of the oocytes and sex steroid productions. Another organs are the fallopian tubes, which is basically the main site for fertilization. Here we have the fimbriae, the uterus, the cervix at the bottom of the uterus, and then the vagina. Okay, so now we're going to be talking about the menstrual cycle. During the first phase, the follicular phase, the primary follicle is growing. As it grows, it develops a second follicle. As it grows, it develops the mature follicle. Around the 14th day of the menstrual cycle, the mature follicle ruptures and releases a secondary oocyte. And this is called ovulation. After ovulation, it becomes an endocrine gland called the corpus luteum. As the luteal phase goes on, the corpus luteum regresses. Okay, so now we're going to talk about the process of fertilization. As the head of the sperm cell encounters the corona radiata of the secondary oocyte, the acrosomal vesicle ruptures, and the sperm cell digests a path for itself by action of the enzymes released from the acrosome. Right there. When the plasma membrane of the sperm contacts the plasma membrane of the ovum, they become continuous, and the nucleus of the sperm cell moves into the cytoplasm of the ovum. When the sperm enters the oocyte, calcium is released from the endoplasmic reticulum. It causes a rise in cytoplasmic calcium that spreads from point of entry to the opposite pole of the oocyte, creating a calcium wave. It also prevents other sperm from entering the oocyte by forming a fertilization envelope.